It's not a primary source of energy. And that's because the vast majority of the cells in your body either don't have the ability to take it up, what we call transport, or they don't have the appropriate enzyme to phosphorylate that fructose, right? Or it's just that those cells are so dependent and reliant on phosphorylating glucose that the idea that they would also take up fructose is probably negligible. So, I mean, and probably alluding to past guests too, one of the main things that fructose is converted to is lipids. And so that is when you consume too much fructose, that is a case where you can then convert it into lipid storage. And those lipids can be used. It's obviously harder to break those down, but you can use those for energy use. And then the other way is what we call the polyol pathway. And this occurs in your liver, where you're literally converting. It's kind of like gluconeogenesis, but it's gluconeogenesis from fructose. So you're basically making glucose from fructose via this polyol pathway through what's called sorbitol intermediate, right? And those two are interconvertible, glucose to fructose and back again. And so a lot of people who, even in the brain, people have observed, they thought if any fructose is even getting to the brain, it's probably just converted to glucose.
I see. So there are situations in which, although fructose is not used itself as a fuel source in many cells most of the time, fructose can be used in certain contexts to create more glucose.
Yeah.
So what about macrophages? Are they able to suck up fructose from the environment? Do they have those transporters?
Yeah. So we'll get onto this, but one of the first things I always like to do when I come across something interesting protein is just look at, data sets that have basically sequencing of every macrophage. And it turned out that if you look at the primary transporter for fructose, GLUT5 or SLC2A5, it's not expressed in any macrophage in humans or mice or rats or monkeys other than one. And that's the brain macrophage called microglia.
And so there's many.
Yeah, go ahead.
Yeah. So as we said earlier, there's many different types of macrophages. Many of them are specialized for certain tissues. You're saying that none of the macrophages have this transporter that allows you to grab fructose from the environment and internalize it, except the macrophages that live in the nervous system. And so let's talk about microglia for a minute. So these are the macrophages that live in the nervous system. There's a lot we could go into here. I don't want to go too deep into the weeds, but can you give us just a basic simplified sense for how were people thinking about microglia up until, not that long ago? And what have we started to learn recently about the totality of what they're doing in the brain?
Yeah. So, you're alluding to a really interesting point, which was up until I think many groups would credit the Barra's lab in California. Up until that point, neuroscientists generally thought microglia weren't really that important of a cell. They were just a small glia cell. And it wasn't, they probably did something, but it wasn't super clear what they did. But the main focus was, you have astrocytes that are dealing with a lot of the toxic substances. You have oligodendrocytes that are important for myelinating and remyelinating, et cetera. And then you have neurons that are the money, right? That's really where the action is in the brain.